I welcome you all to the session on reproduction of plants. I'm Dr. Beema Jainab, Assistant Professor from the Department of Plant Biology and Plant Biotechnology, working in Justice Bashir Ahmed Syed College for Women, Chennai. In this session, I'm going to highlight about the tips for teachers and sexual reproduction in plants. So let's move on to the tips for teachers. The activity. Directions are provided to the students to conduct activities in order to explore, enrich the concept. Infographics, visual representation of the lesson to enrich learning. Evaluation, access students to pass, think and check their understanding. Content, to motivate the students to further explore the content digitally and take them into virtual world. Now we are moving on to the topic sexual reproduction. Sexual reproduction involves the production and fusion of male and female gametes. The former is called gametogenesis and the latter is the process of fertilization. Alga and bryophytes reproduce by the production of gametes which may be motile or non-motile depending upon the species. In alga, external fertilization takes place whereas in higher plants, internal fertilization occurs. First, going on into that, we have to know what is a flower. Flower is a condensed shoot. It is meant for reproduction. A flower possesses 4 whorls. They are the calyx, corolla, androecium and gynoecium. Androecium and gynoecium are essential organs. So all these parts you can see on to this picture. The next, we are moving on to the stages of process or changes involved in sexual reproduction of higher plants. Here there are 3 important steps. They are the pre-fertilization structure and events. Then comes the fertilization and post-fertilization changes. Now we are going to consider pre-fertilization structure and events. The hormonal and structural changes in plant lead to the differentiation and development of floral primordium. What is differentiation? Animals and plants produced by sexual reproduction begin life as a single cell, a fertilized egg or zygote. These cells must divide by mitosis to produce a multicellular organism. Floral primordium. It is a group of cells or a tissue in an angiosperm or a flowering plant which gives rise to a flower. It is a tissue or a colony of cells that trigger the formation of an organ before its formation. The next one is the male reproductive part, androecium. Androecium is made up of stamens. Each stamen possesses an anther and a filament. Formation of haploid microspores from diploid microspore mother cell through meiosis is called microsporogenesis. The primary sporogenous cells directly or may undergo a few mitotic divisions to form sporogenous tissue. The last generation of sporogenous tissue functions as microspore mother cells. Anther bears pollen grains which represent the male gametophyte. In some plants, all the microspores in a microsporangium remain held together called pollinia. Example for it, Calotropis. Pollinia are attached to a clamp or clip-like sticky structure called corpusculum. The filamentous or thread-like part arising from each pollinium is called retinaculum. The whole structure looks like letter Y and that is known as translator. The next one is the development of the anther. A very young anther develops as a homogeneous mass of cells Surrounded by an epidermis, during its development, the anther assumes a four-lobed structure. You can see in this picture. In each lobe, a row or a few rows of hypodermal cells becomes enlarged with conspicuous nuclei. Thus function as archisporium. The archisporial cells divide by periclinal divisions to form primary parietal cells towards the epidermis and primary sporogenous cells towards the inner side of the anther. The primary parietal cells undergo a series of periclinal and anticlinal division, and it forms 2 to 5 layers of anther walls, composed of endothecium, middle layers, and tapetum from periphery to center. The next one, we are going into the structure of a mature anther. In the transverse section of mature anther reveals the presence of anther cavity surrounded by an anther wall. It is bilobed, each lobe having 2 theca, dithecous. A typical anther is tetrasporangiate. First, if you take up the anther wall, the mature wall consists of the following layers. Epidermis, it is a single layered and protective in function. The cell undergoes repeated anticlinal divisions to cope up with the rapidly enlarging internal tissues. Endothecium, it is a single layer of radially elongated cells found below the epidermis. The inner tangential wall develops bands of alpha cellulose. The cells are hygroscopic. The cells along the junction of the 2 sporangia of an anther lobe lack these thickenings. This region is called stomium. This region, along with the hygroscopic nature of endothecium, helps in the dehiscence of the anther at maturity. You can see in this picture the diagrammatic view as well as that is viewed under the microscope. The next layer is the middle layers. It is about 3 layers of cells next to the endothecium constitute the middle layers. They are generally ephemeral. They disintegrate or get crushed during maturity. Tapetum is the most important layer. It is the innermost layer of the anther wall and attains its maximum development at the tetrad stage of microsporogenesis. It is derived partly from the peripheral wall layer and partly from the connective tissue of the anther lining the anther locule. Thus, the tapetum is dual in origin. It nourishes the developing sporogenous tissue, microspore mother cells and microspores. The cells of the tapetum may remain uninucleate. It also contributes to the wall material sporopollenin, pollen kit, tryphin and number of proteins that control incompatibility reaction. Tapetum also controls the fertility or sterility of the microspores or pollen grains. There are 2 types of tapetum based on its behavior. The first one is the secretory tapetum. It can be called as parietal, glandular, cellular. The tapetum retains the original position and cellular integrity and nourishes the developing microspores. Invasive tapetum is otherwise known as periplasmodial. The cells lose their inner tangential and radial walls and the protoplast of all tapetal cells coalesces to form a periplasmodium. The next, we are moving on to the functions of the tapetum. It supplies nutrition to the developing microspores. It contributes sporopollenin through Ubich bodies, thus plays an important role in pollen ball formation. The pollen kit material is contributed by tapetal cells and it's later transferred to the pollen surface. Exine proteins responsible for rejection reaction of the stigma are present in the cavities of the exine. These proteins are derived from tapetal cells. The next one is the anther cavity. The anther cavity is filled with microspores in young stages or with pollen grains at maturity. The meiotic division of microspore mother cells gives rise to microspores which are haploid in nature. The next is connective. It is the column of sterile tissue surrounded by the anther lobe. It possesses vascular tissues. It also contributes to the inner tapetum. Next is the microspores, or the intermediate product of meiosis of the microspore mother cell, whereas the pollen grain is derived from the microspore. The microspores have protoplast surrounded by a wall which is yet to be fully developed. The pollen protoplast consists of dense cytoplasm with a centrally located nucleus. The wall is differentiated into two layers, inner layer called intine and outer layer exine. Intine is thin, uniform, and it is made up of pectin, hemicellulose, cellulose and callose together with proteins. Exine is thick and it's made up of cellulose, sporopollenin and pollen kit. The exine is not uniform and it is thin at certain areas. When these thin areas are small and round, it is called germ pores, or when elongated, it is called furrows. It is associated with the germination of pollen grains. The sporopollenin is generally absent in germ pores. The surface of the exine is either smooth or sculptured in various patterns, rod-like, grooved, warty, punctuate, etc. The sculpturing pattern is used in the plant identification and classification. The pollen grains are having different shapes. They are the globose, ellipsoid, fusiform, lobed, angular or crescent. The size of the pollen also varies from 10 micrometers in Myosotis to 200 micrometers in the members of the family Cucurbitaceae and Nyctaginaceae. The wall material sporopollenin is contributed by both pollen cytoplasm and tapetum. It is derived from carotenoids. It is resistant to the physical and biological decomposition. It helps to withstand high temperature and it's resistant to strong acid, alkali and enzyme action. Hence, it preserves the pollen for long periods in fossil deposits and it also protects pollen during its journey from anther to stigma. What is a pollen kit? It is contributed by the tapetum and colored yellow or orange, and it's chiefly made up of carotenoids or flavonoids. It is an oily layer forming a thick viscous coating over the pollen surface that will attract the insects and it protects the damage from UV radiation. The next one is the development of the male gametophyte. The microspore is the first cell of the male gametophyte and it is haploid. The development of the male gametophyte takes place while they are still in the microsporangium. The nucleus of the microspore divides to form a vegetative and a generative nucleus. I hope so. You can easily observe it in this picture. A wall is laid around the generative nucleus, resulting in the formation of two unequal cells. A large irregular nucleus bearing with abundant food reserve that you call it as vegetative cell and a small generative cell. At this two-cell stage, the pollens are liberated from the anther. In some plants, the generative cell again undergoes a division to form two male gametes. In these plants, the pollen is liberated at three-celled stage. In 60% of the angiosperms, pollen is liberated in two-cell stage. Further, the growth of the male gametophyte occurs only if the pollen reaches the right stigma. The pollen, on reaching the stigma, absorbs moisture and swells. The intine grows as pollen tube through the germ pore. In case the pollen is liberated at two-cell stage, the generative cell divides in the pollen into two male cells, that is the sperms, after reaching the stigma or the pollen tube before reaching the embryo sac. The next one is the female reproductive part, gynoecium. It represents the female reproductive part of the flower. It represents one or more pistils of a flower. The pistil refers to the ovary, style and stigma. It is derived from a carpel. The word ovary represents the part that contains the ovules. The stigma serves as a landing platform for the pollen grains. The style is an elongated slender part beneath the stigma. The basal swollen part of the pistil is the ovary. The ovules are present inside the ovary cavity, locule on the placenta. The carpel arises as a small papillate outgrowth of a meristematic tissue from the growing tip of the floral primordium. It grows actively and soon gets differentiated into ovary, style and stigma. The ovules or megasporangia arise from the placenta. The number of ovules in an ovary may be 1, paddy, wheat and mango, or many in the case of papaya, watermelon and orchids. Structure of the ovule, that is the megasporangium, and it is produced by 1 or 2 covering called integuments. A mature ovule consists of a stalk and a body. The stalk or the funiculus is present at the base and it attaches the ovule to the placenta. The next one is the point of attachment of the funicle to the body of the ovule, that we call it as hilum. It represents the junction between the ovule and funicle. In an inverted ovule, the funicle is adnate to the body of the ovule, forming a ridge called raphe. The body of the ovule is made up of a central mass of parenchymatous tissue called nucellus, which has large reserve food materials. The nucellus is enveloped by 1 or 2 protective coverings called integuments. Integument encloses the nucellus completely except at the top where it is free and forms a pore called the micropyle. You can see the micropyle in the picture. The ovule with 1 or 2 integuments are said to be unitegmic or bitegmic ovules respectively. The basal region of the body of the ovule, where the nucellus, the integument and the funicle meet or merge, that end you call it as chalaza. That is also as depicted in the picture view. The next one, the embryo sac or the female gametophyte. It is a large, oval, sac-like structure in the nucellus towards the micropylar end called as embryo sac. It develops from the functional megaspore formed within the nucellus. In some species, the inner layer of the integument may become specialized to perform the nutritive function for the embryo sac, and we call it as endothelium or integumentary tapetum. This example is found in Astraceae. There are 2 types of ovule based on the position of the sporogenous cell. If the sporogenous cell is hypodermal with a single layer of nucellar tissue around it, we call it as tenunucellate type. This type of ovules have a very small nucellus. Ovules with subhypodermal sporanginous cell is called crassinucellate type. This type of ovules have fairly large nucellus. Group of cells, when it is found at the base of the ovule between the chalaza and embryo sac, is called hypostase. And the thick-walled cells found above the micropylar end, above the embryo sac, that we call it as epistase. The next one is the types of ovules. The ovules are classified into 6 main types based on its orientation, form and position of the micropyle with respect to funicle and chalaza. Orthotropous. In this type of ovule, the micropyle is at the distal end and the micropyle and the funicle and the chalaza lie in one straight vertical line. You can see it in the picture. Examples are Piperaceae, Polygonaceae. The another type is anatropous. The body of the ovule becomes completely inverted so that the micropyle and the funiculus come to lie very close to each other. This is the common type of ovules found in dicots and monocots. That also you can see it in the picture. The third type in the picture is hemianatropous. In this, the body of the ovule is placed transversely and at right angles to the funicle. Example will be primulaceae members. The next type of ovules are campylotropous. Here the ovule at the micropylar end is curved and more or less bean shaped. The embryo sac is slightly curved. All the three, hilum, micropyle and chalaza, are adjacent to one another, with the micropyle oriented towards the placenta. Members of Leguminosae have this type of ovule. The next one is amphitropous. The distance between hilum and chalaza is less here. The curvature of the ovule leads to horseshoe-shaped nucellus. Example, the family members of Alsmataceae. Cercinotropous. Here the funiculus is very long and surrounds the ovule. Here the family members of Cactaceae possess this type of ovule. And we are moving on to the megasporogenesis. The process of development of megaspore from a megaspore mother cell is known as megasporogenesis. Here the ovule develops a single hypodermal cell in the nucellus, becomes enlarged and function as archisporium. Then we are moving on to the types of embryo sac based on the number of megaspore. We have monosporic, bisporic and tetrasporic. Monosporic is the 4 megaspores are formed usually at the chalazal 1 is functional and other 3 megaspores degenerate. The functional megaspore forms the female gametophyte or embryo sac. This type of development is called monosporic development. This you find it in Polygonum. Bisporic, one of the 4 megaspores formed if 2 are involved in embryo sac formation. The development is known as bisporic. Example will be Allium cepa. Tetrasporic, if all the 4 megaspores are involved in embryo sac formation, the development is called tetrasporic. Example will be Peperomia. The next one, the development of monosporic embryo sac. The functional megaspore is the first cell of the embryo sac or the female gametophyte. The megaspore elongates along the micropylar chalazal axis. The nucleus undergoes a mitotic division. Wall formation does not follow the nuclear division. A large central vacuole now appears between the 2 daughter nuclei. The vacuole expands and pushes the nuclei towards the opposite poles of the embryo sac. Both the nuclei divide twice mitotically, forming 4 nuclei at each pole. At this stage, all the 8 nuclei are present in a common cytoplasm, free nuclear division. After the last nuclear division, the cell undergoes appreciable elongation, assuming a sac-like appearance. This is followed by cellular organization of the embryo sac. Of the 4 nuclei at the micropylar end of the embryo sac, 3 organize into an egg apparatus. The 4th one is left free, the cytoplasm of the central cell, as the upper polar nucleus. Now we are moving on to the development of monosporic embryo sac. 3 nuclei of the chalazal end form 3 antipodal cells, whereas the 4th one functions as the lower polar nucleus. Depending on the plant, the 2 polar nuclei may remain free or may fuse to form a secondary nucleus central cell. The egg apparatus is made up of central egg cell and 2 synergids, one on each side of the egg cell. A clear picture represents the monosporic embryo sac. Here you can see the synergids that secrete chemotropic substances that help to attract the pollen tube. The special cellular thickening called filiform apparatus of synergids helps in the absorption conduction of nutrients from the nucellus to embryo sac. It also guides the pollen tube into the egg. Thus, a 7-celled with a 8-nucleated embryo sac is formed. That is what you are seeing in the picture. Thank you so much for your patience listening. Hope so you have understood about the sexual reproduction, especially on the gynoecium as well as the andrecium.